M phase of the cell cycle stands for mitosis. And mitosis really refers to the division of one nucleus to form two new nuclei, each of which contain genetic material which is identical to each other and to the original nucleus or the original parent cell. You should already be familiar with the basic stages of mitosis. I'm going to review them fairly quickly, but I've also asked that you read carefully the parts in your textbook which describes the different phases of mitosis to let you know what is happening both to the chromosomes and to the cytoskeleton and the rest of the cytoplasm during mitosis. Interphase is a period of time prior to mitosis. It's during interphase that DNA replication takes place and the cell begins to prepare for mitosis. And this can include creating the appropriate proteins and enzymes that will be necessary for mitosis. These images you'll be seeing were taken by Conley Reeder, who's a research scientist who studies mitosis. The cells have been triple stained. First with a blue dye that labels the DNA, and you can see the nucleus in the middle of this image. The green labels microtubules, and the red labels intermediate filaments. In interphase, you can see that the chromosomes are spread out. They're existing as euchromatin and heterochromatin in the nucleus. They have not yet condensed to form the kinds of chromosomes that we actually see in mitotic cells. Prophase marks the beginning of mitosis. The chromosomes start to fold up increasingly around additional proteins inside the nucleus in order to become visible, as you can see in this image here. Throughout the entire stage of prophase, the nuclear envelope remains intact. Outside of the nucleus, the cytoskeleton has begun the changes associated with preparing to divide the chromosomes. As you can recall, during S phase, the centrioles duplicated. It's during prophase that these two centriole pairs begin to separate. Each of these pairs of centrioles act as microtubule organizing centers, and so the cell now has two microtubule organizing centers, and you can see that microtubules are nucleating around both of these centers and generating what are known as asters. I hope that you can remember the polarity of these microtubules, so where the plus end and the minus end would be. If you think about mitosis, it's in anaphase that the sister chromatids will be separated and moved to opposite poles of the cell. But if you look at an interphase cell, shown here, there aren't two poles to this cell. So one of the roles of this massive rearrangement of the cytoskeleton in mitosis is to create two poles inside the cell, or two ends inside the cell, so that the sister chromatids, once they separate, can be moved to opposite ends of the cells. This creation of this polarity inside the cell begins in prophase, with the separation of the two centriole pairs. This moving apart appears to be generated by the activity of motor proteins that push the microtubules in the two microtubule organizing centers away from each other. If you look at the chromosomes at this stage, you can see that they look like long sort of worm-like structures, and they look like each one of these worms is a single element. However, each one consists of two sister chromatids. The reason why they appear as one is that the sister chromatids are being held closely together by the cohesins. The condensation of the chromosomes that one sees in prophase is the result of the activity of additional proteins inside the nucleus, including condensins. These condensins help to loop or form rings and loop structures in the DNA, which helps it fold up more tightly. Not shown here is going to be the activity that's happening at the centromeres. So you'll remember these were stretches of DNA. There's usually a single centromere in one chromosome. It's around the centromere that additional proteins are going to start to be recruited and start to form a new structure, which is known as the kinetochore. We'll talk a little bit more about the kinetochore shortly. The transition between prophase and prometaphase is marked by the loss of the nuclear membrane. So the nuclear membrane essentially starts to break down. And by doing so, it releases the chromosomes which have been condensing into the cytoplasm. And this allows the chromosomes to interact with the microtubules in the cytoplasm to start form connections and begin to build the spindle. These connections between the chromosomes and the microtubules to form spindle fibers will be mediated by the structure known as the kinetochore. So the centromeres and the kinetochores are not the same thing. The centromere is a region of DNA, and the kinetochore is a protein structure which is built at the centromere. So if you were to look at this area, this region here, which is the centromere region, and look at a very high magnification, you can actually see the structure of the kinetochore. It's a layered-like structure, and you can see it right here. So here's a chromosome in black. This is a high magnification electron micrograph. And you can see that there are different layers to the kinetochore. There's a darker layer closest to the chromosomes. There's a light layer in between, like a sandwich, and then there's an outer layer here, which is again slightly dark. And you can see the microtubules which have been attached to the kinetochore and are emanating out from the kinetochore. Now here we see that there are microtubules going in both the top direction and the bottom direction. And that is because there are two kinetochores here. Each sister chromatid has a kinetochore. So there's a sister chromatid sort of outline if you sort of follow my cursor here up at the top, and a second sister chromatid down at the bottom. They're being held together by these cohesins, so you can't see them as separate entities. But you can tell that they're separate entities because they each have a centromere and a kinetochore structure. During prometaphase then, the microtubules make attachments with the kinetochores, and they start to move the chromosomes back and forth within the spindle. You can see here the green microtubules forming two poles, and the chromosomes are laid out in between them, moving back and forth. Some chromosomes have been pulled right up to one of the poles or the other, and other ones are caught in between. This pushing and pulling continues, and it's due to the activity of motor proteins acting on the microtubules to move the chromosomes back and forth until eventually they come to rest in the middle of the cell, or in the middle of the spindle at what is known as the metaphase plate. When that happens, the cell transitions into metaphase. At metaphase, the spindle is at its most mature form. You have two clear poles at either end of the cell. The chromosomes are all aligned in the middle. And the reason why you still see all this fuzziness of the chromosomes sticking out is because these are the chromosome arms. And what's actually aligned at the metaphase plate are the kinetochores, because the kinetochores are the parts of the chromosomes that are directly attached to the spindle. The microtubules that connect the kinetochores to the poles of the spindle are known as kinetochore microtubules. Typically, once metaphase is reached, it does not last very long, and the cell very quickly moves into the next stage, which is anaphase. Before we leave metaphase, however, let's take a look at the different kinds of microtubules inside the spindle. Some of the microtubules are directly attached to kinetochores, and these form what are known as kinetochore microtubules. They are also sometimes called kinetochore fibers. In addition to the microtubules connecting kinetochores to poles, there are a number of other microtubules that simply form the rest of the spindle. They'll be attached to one pole and extend towards the metaphase plate. These are known as non-kinetochore microtubules. The non-kinetochore microtubules from both poles interact and intercalate with each other around the area of the metaphase plate. And finally, there are additional microtubules that extend from the two centriolar regions or from the microtubule organizing center in all directions, and they tend to be fairly short, and they're connecting upwards towards the cell surface or the cell membrane, and these are known as astral microtubules. It's during anaphase that the sister chromatids actually separate. So in the transition from metaphase to anaphase, the cohesins are actually broken down. They can no longer keep the sister chromatids attached, and the cell moves into anaphase. The chromosomes begin to move towards the pole. And that poleward movement appears to be due to two fundamental activities. One is the activity of motor proteins, acting with the kinetochore microtubules, and the second element is, of course, the depolymerization of the kinetochore microtubules themselves, because as the chromosomes move poleward, these kinetochore fibers or kinetochore microtubules must necessarily be shortening. When I was a graduate student, I actually studied anaphase in meiotic cells, and there are a number of different experiments that were carried out. Your textbook describes one of them, which provides evidence that the depolymerization of the microtubules probably happens in the kinetochore region. In the cell type that I was studying as a graduate student, the evidence that we obtained suggested that depolymerization was actually happening at both ends of the kinetochore microtubules. Once the chromosomes hit the poles, we move into the final stage of mitosis, which is known as telophase. The main role of telophase is to transition the cell out of mitosis and back into interphase. When the chromosomes reach the poles, that sets off additional changes both in the cytoskeleton of the cell and changes in the chromosomes. The chromosomes begin to decondense, the nuclear membrane reforms, and the cytoskeleton returns back to a normal interphase cytoskeleton. It is also often during telophase or late anaphase when the cell begins cytokinesis. And before I discuss cytokinesis, I just wanted to point out that the different stages of mitosis we've just discussed and the images that you've been watching are from animal cells. And first of all, different animal cells may have slightly different types of mitoses. It doesn't always look exactly the same. And as I've mentioned before, mitosis does vary somewhat in different types of organisms. In plant cells, for example, they lack centrioles. They still contain microtubule organizing centers, but they're more spread out and there seem to be more of them around the nuclear membrane. When the nuclear membrane breaks down, these microtubules can attach to the kinetochores and they will still form a bipolar spindle. So there are still two ends. They create two ends in this cell. However, you can tell that without the centrioles, the microtubules or the kinetochore fibers do not create this very focused pole as we see in animal cells. Nevertheless, when the sister chromatids separate, they're able to move to two opposite directions in the cell, or the two sort of broader, less focused poles, and move into telophase. Somewhere usually in late anaphase and into telophase, the actual cell will divide in two. In other words, cytokinesis occurs. How it happens varies again by cell type, and we'll just look at the differences between animals and plants. In animal cells, you'll remember that they lack a cell wall, and therefore the cell membrane is much more flexible. And so in animal cells during cytokinesis, there is a tight ring of actin that forms just below the cell membrane in the area between the two spindle poles. It's essentially where the metaphase plate had originally formed. These actin filaments, with the help of myosin, will basically pinch the cell into two. And here you can see this cleavage furrow, and if you were to look at the bottom of this furrow down here, you'll see a tremendous amount of actin, lots of actin bundles, and this pinching in is happening with the activity of myosin and actin together. In the case of plant cells, they have to work within the boundaries of a rigid cell wall, and so the cytoplasm itself doesn't really pinch in two. Instead, new membrane is actually created in between the two sets of chromosomes, or again, essentially where the metaphase plate was originally located. This is formed by the fusion of large numbers of membrane-bound vesicles. During the latter stages of anaphase and telophase, you still have microtubules, these would be the non-kinetochore microtubules, projecting into the spindle area towards the metaphase plate, and they start to carry vesicles along them towards the metaphase plate. And you should be able to tell me whether they're moving towards the plus or the minus end of the microtubules. When the vesicles reach the metaphase plate area, or the midway between these two groups of chromosomes, they find each other and begin to fuse. And in fusing, they actually create new membrane here, sort of like a giant bubble, which expands across the cell and will eventually meet up and fuse with the cell membrane on either side of the cell. So eventually you'll have a new membrane on this side and a new membrane on that side, creating two new cells, and what is deposited in between those two cell membranes will be the materials to form the cell wall. This area here where the new membrane is forming is known as the cell plate. And this is again a very high magnification electron micrograph of a plant cell at the end of division, and you can see the vesicles that are forming here at the cell plate and creating this new membrane as they fuse together. And that pretty much finishes up our description of mitosis and cytokinesis. Some terminology that you should be comfortable with are listed here, so make sure you can define each one of these and can distinguish between each one of these terms.